Hi there. This is one more short video to help introduce the idea of cellular chassis within the context of synthetic biology. So I'm Natalie Coldell and I will talk through these slides, but I want to credit and give tremendous thanks to my friend Barry Canton at Ginkgo Bioworks, who initially developed most of these slides and gave this talk. And as you'll see, I'll refer to his work throughout the talk. But where I'd like to start is in case you have done some reading and some thinking about another of the BioBuilder activities called Oh That Smell, which is based on the Eau de Coli project from the MIT iGEM team in 2006. And I want to start here just to remind you that in addition to the really interesting genetic elements that they brought into this system to make the bacteria smell like bananas or smell like wintergreen, they also did a modification to the host cell that was carrying those genetic programs, that is to the chassis that was driving that genetics that went in, the plasmids that went in. And the modification that they made was to remove the stinky indole smell by mutating a tryptophanase on the genome of that E. coli. So that is an example of chassis engineering that maybe you're already familiar with. So with that as a perhaps starting place, let me back up just a little bit and start where Barry very thoughtfully introduced these ideas of chassis properties. I'm going to start at the bottom of the slide and talk about niche utility because in a way I already did talk about niche utility. The option of removing the smell from E. coli is a niche property that many chassis would not need, but that perhaps is extremely useful in certain cases, like the banana smell project. There are other niche utilities for chassis. Sometimes you'll want the cell to be human-like, sometimes you might want it to be not alive and just a sack of biochemicals. Sometimes the intellectual property status is going to be critically important to whether you can move forward on a project or not. So those are important considerations, but for most of this short video lesson, what I'd like to focus on is the general utility of chassis, things that we would want the cells to be. Certainly at the top of the list is safe, and I'll end some of these slides by discussing how you might engineer safety. Then you want them to be as simple as possible, perhaps because we can understand simpler things, that the more complex things get, the more unexpected outcomes there might be. And you would want them to be engineerable. They may need a genetic system where you can express unusual genes in them. You want them to be things you can manipulate rationally and reliably. So the first approach to engineering a chassis will be by starting with something close. So what I mean by that is if you have a bunch of organisms you would like to engineer called organism 1, 2, 3, dot, 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 all the way to engineered organism N. So a whole slew of these. One way to get to these final organism products would be to find a wild type organism, something that nature has provided that is close, that is something akin to what the engineered organism is you're trying to build, and then move that wild type organism towards the engineered organism you would like. So there are many good examples of this as a successful approach, this nearest wild type organism approach. Here's an example from ARPA-E, which is the Advanced Research Project Agency focused on energy. And it's a proposal which uses an organism called Ralstonia eutropha to make energy alcohols, branched chain alcohols. So the really neat thing about Ralston eutropha is that you can teach this microbe to make fuel, is what this news release says about it. What the organism can do is it takes carbon from the environment and stores it as a polymer. That's something the organism does well naturally. In fact, if it's under stress, some of the numbers I read were that 80% of its dry weight becomes stored as carbon stored molecules, branched molecules. So what this group led by Tony Sinsky at MIT did was to port in a number of genes that would enable this very nifty chassis, Ralston eutropha, to convert some of the cell's pyruvate stores into isobutanol and 3-methyl-1-butanol. So you can see at the bottom of the slide on the right that if you look over a couple of months, so out to 50 days or so, you get increasing titer of branch chain alcohol production in terms of grams per liter accumulating over time. So these cells, which naturally like to store carbon, can store these particularly useful branch chain alcohols by engineering this pathway into them. So that's an example of the nearest wild type organism approach to chassis engineering. An alternative approach is to start perhaps with a standard chassis. So the idea here is that if you need a whole slew of engineered organisms, 1, 2, 3, 4, 5, down to N, you could get to all of them from some starting material, a starting chassis. And if this seems far-fetched, the ability to take one cell and turn it into something that can produce fuel, and take that same cell and engineer it so that now it can produce food, and something else so that now it could produce medicine, you may be right. That is a stretch. And so maybe rather than starting with one chassis, we may want to have a small menu of starting standard chassis that could be engineered and further developed to become the organisms that we would like them to be. And so this alternative example is one that Ginkgo Bioworks is focused on, where they do try to engineer organisms in a forward-looking way. So take a chassis and make it into an organism that you would like to produce X, Y, or Z. They have a ARPA-E proposal as well, and this one also to make fuel. This is an electrofuel isooctane, but not starting with Ralston, which is already good at storing carbon, but starting with E. coli, a very standard laboratory workhorse that will then be engineered to produce electrofuels. So those are the two contrasting approaches. You start with something close and turn them into engineered organisms 1 through N, but you need, in that case, starting organisms 1 through N to reach your endpoint. The alternative is you start with a standard chassis or perhaps a small menu of standard chassis and use those to further develop organisms 1 through N. Each idea has its benefits and drawbacks. Obviously, one maybe further from where we currently stand in the field right now, but it would be important to know what would make chassis engineerable and how we could move forward on them if we wanted a small menu of standard chassis to engineer. In addition, while we're thinking about engineering these chassis, it's important to always consider the safety questions around chassis. And well, here are two ideas that Barry spotlighted to make these chassis more safe. The second of these two, make your chassis weak in particular environments, is something that oftentimes will happen even by not trying, that is by loading in some of these features or by stripping away from these cells things that are useful for the cell to survive in the wild. It automatically weakens that cell versus the wild type cells that we might find. And so should that chassis escape, its likelihood of surviving outside of a laboratory environment diminishes. And then there are ways to further ensure that escaped chassis, if they should accidentally be released, would not be able to survive in an environment that, say, lacks a particular nutrient. The top idea that make your chassis DNA orthogonal to natural species is a really interesting one to think about as an engineer. The example that was published in 2011 of recoding E. coli so that one of the stop codons is no longer used and freeing up that stop codon to perhaps encode something else is a very interesting article that was published, as I say, in Science in 2011. This E. coli project used a genome engineering technique called CAGE and MAGE, a combination of the two, to engineer the genome so that this stop codon, TAG, is no longer encoding a stop in the E. coli. And what that means is that any DNA that might come into that bacterial cell that uses the TAG as a stop codon, it couldn't be read by this recoded E. coli. And so new DNA that might come into it would not be meaningful in this modified chassis. So the experiment that BioBuilder provides for exploring differences in chassis is the What a Colorful World experiment that allows you to try your hand at bringing genetic programs into different cellular chassis, two types of E. coli, a B type and a K12 type of E. coli, very close cousins of one another, and to ask whether these genetic programs behave identically or differently in these two cells. And we think of cells in science often as a very generic term, but in reality, of course, cells have very different properties, one from the other. And so this is an experiment that would allow you to explore that. So that is the end of this short lesson. I hope it's been interesting and helpful to you as you think about the host that runs these genetic programs, which in engineering terms is often called the chassis, and the variations that you might want to see to them.